The astonishing ability of shrews to shrink their brains during winter is not due to the loss of brain cells, but rather the loss of water, a discovery that could revolutionize how we understand brain diseases in humans. But here's where it gets controversial. Unlike in humans, where brain shrinkage is a hallmark of neurodegenerative diseases like Alzheimer's, shrews manage to reduce their brain size seasonally, without suffering any functional damage. This phenomenon, known as Dehnel's phenomenon, has mystified scientists for decades, but new research offers fresh insights that might even pave the way for future medical breakthroughs. Researchers from the Max Planck Institute have used cutting-edge MRI technology, the same imaging method doctors use to scan human brains, to study live common shrews throughout the changing seasons. What they uncovered was remarkable. Shrews lose about 9% of their brain volume in winter. But crucially, this shrinkage comes from water leaving the brain cells rather than the cells themselves dying. This sets them starkly apart from humans, where water loss within brain cells is usually a sign of damage leading to cell death. Cecilia Baldoni, the lead author of the study and a postdoctoral researcher, remarked, "Our shrews lost almost a tenth of their brain size during the colder months, but none of their brain cells died. Instead, the cells simply lost water. Astonishingly, they even observed an increase in the number of brain cells during this period, flipping the usual narrative of shrinkage leading to decline on its head." This discovery is not just fascinating from a biological standpoint. It could have major implications for human health. Brain shrinkage in humans is commonly linked to devastating diseases such as Alzheimer's, Parkinson's, and other neurodegenerative conditions. According to John Nealand, an expert in human brain diseases, the parallels between brain shrinkage caused by disease in humans and the seasonal shrinkage in shrews are striking. But crucially, shrews have the ability not only to survive this shrinkage, but also to reverse it. A particularly interesting part of this puzzle involves a specific protein called aquaporin 4, which regulates water flow in and out of brain cells. The researchers found that this protein plays a key role in shrews by helping move water out of brain cells during winter. And intriguingly, this same protein is found in elevated levels in diseased human brains. Could studying shrews teach us how to manipulate this protein for therapeutic purposes? That's the burning question. Dehnel's phenomenon, the reversible shrinking and regrowth of the brain, is an extremely rare trait found in only a few mammals like European moles, stoats, weasels, and some species of shrews. Among these, the common shrew is the best studied. Through high-resolution MRI scans of wild shrews caught in summer and re-examined in winter, scientists tracked these seasonal changes non-invasively, allowing them to compare individual brains over time and establish that water loss is the key factor behind brain shrinkage. One of the biggest puzzles was how shrews managed to cope with a smaller brain while still performing complex behaviors, like hunting for food and escaping predators during harsh winters. "It's quite unbelievable," says Baldoni. "Despite their brains shrinking by nearly 10%, similar to the brain loss seen in Alzheimer's patients, shrews show no decline in function." Nealand echoes this, emphasizing the stark contrast between the debilitating impact of brain shrinkage in humans and the shrews' seemingly effortless adaptation. The study revealed fascinating regional differences in brain shrinkage. While most parts of the brain reduced in size and showed clear signs of water shifting from inside to outside the cells, critical areas like the neocortex and cerebellum maintained their water balance more stably. These regions are responsible for higher cognitive functions such as memory and motor control. Baldoni likens this to adjusting the heat in a home. The shrews preserve heating in the essential brain rooms while scaling back energy in less critical areas during the winter. This nuanced understanding opens new doors for ecological and behavioral research. Scientists are now eager to investigate how this brain shrinkage affects shrews' cognitive abilities across seasons. Can they navigate and coordinate just as well in winter with smaller brains? This remains an intriguing question. For medical science, the implications are profound. Many human neurological disorders, including multiple sclerosis, Parkinson's disease, ALS, and Alzheimer's, involve brain shrinkage due to water loss, which currently only progresses and never reverses. No existing treatments can halt or reverse brain volume loss, explains Dehnel. But shrews, suffering from a similar brain volume reduction, possess natural biological tools not only to stop but to completely reverse this process. The next challenge for researchers is to unlock how shrews regrow their brains each spring. Decoding this regenerative process could potentially lead to groundbreaking therapies for currently incurable human brain diseases. Dehnel expressed enthusiasm about this future direction, calling it the most exciting prospect I can imagine in neurological research. But here's where the controversy lies. Should we view the shrew's brain shrinkage strictly as a natural adaptive mechanism, or could it challenge existing paradigms about brain health and disease? Could understanding the shrew's biology force us to rethink the inevitability of brain cell loss in neurodegenerative conditions? What if humans could be taught to mimic such seasonal brain plasticity? We'd love to hear your thoughts. Do you think this small mammal holds the key to a new frontier in brain medicine? Or is it too much of a stretch to compare conditions across species? Join the conversation and share your views.